So what are the most important phases of mitosis? Now in mitosis, the major phases that and in most of the material that you will come across, prophase includes initial stage of metaphase also, which is called prometaphase. So we will directly after prophase talk about metaphase. So the first phase is the prophase. Remember, we are talking about karyokinesis here. Prophase is followed by metaphase, and metaphase is followed by anaphase, which is the third phase, and the last phase, which is telophase. Telophase marks the end of karyokinesis, and of course after that the cytoplasm or the cell divides, and this division of the cell or the cytoplasm is called cytokinesis. So in prophase, you all can see how the centrioles have duplicated and the centrioles are migrating towards the opposite poles, and early mitotic spindle is also forming. And very importantly, when did the centriole duplication happen? The centriole duplication actually happened in the S phase of interphase. Interphase is wrongly called the resting phase. Actually, the cell is not resting, the cell is preparing itself for mitosis. So in interphase you have the S phase, during which the centriole in animal cells undergo duplication. And if you look inside the nucleus you can see chromosomes begin to appear during prophase. During the last stages of prophase or the early stages of metaphase, as you can see in the case of prometaphase, how the chromosomes begin to align at the equator. See, all the chromosomes in the cell will align at the equator in a single line. They don't align in a double line as opposed to what we saw in meiosis. They align in a single line, in a single plane along the equator of the cell, and this is called the metaphase plate. And you can see how the spindle fibers establish connections with the kinetocores of their centromeres. The centromeres are covered with disc-shaped structures, disc-shaped proteins, that is referred to as the kinetochore. And notice that each chromosome has two chromatids associated with it, which are referred to as the sister chromatids, isn't it? So each chromosome that aligns at the equator has a corresponding duplicate chromatid of its own, which is referred to as the sister chromatid. What happens during anaphase? The centromere splits. See, the centromere split. Each chromosome which had two, now if this is a chromosome with two of its sister chromatids, the centromere splits in the center. So as a result, the two sister chromatids are dragged away from each other. So splitting of the centromere and the separation of the sister chromatids happen during anaphase. And lastly, once the chromosomes migrate to the opposite poles, you can see that the chromosome lose their identity as discrete entities here. That is because they start unwinding into the thread-like structure, and you also can see the reappearance of the nuclear membrane. And in this case, you can see how a depression or a furrow appears at the equatorial region, and the furrow keeps deepening. It deepens and finally meets at the center, and it divides the cell into two daughter cells. So this is what the process depicted over here is that of an animal cell where there is formation of a cleavage furrow. So finally we have two daughter cells, and we call mitosis as equational cell division. The reason why we call mitosis as equational cell division is because whatever chromosome complement existed in the mother cell, the same chromosomal complement is there in the daughter cells as well. There is no reduction in the number of chromosomes.